I would give you some examples now of what you can start to do with this data set once we have it in. It's a bit fulfilling to do all this work and then not really be able to ask any questions about the data set. So I would then open this file explore.rmd. It's a our markdown file again. So that's it for the import RMD. And so I would keep that as a separate file, right? That's what I would highly recommend, is that you have basically one R or R markdown file for each step in your project. And really, in my lab, for each experiment, we have a separate file. And I'll come back to that later on. It's good software engineering principles, but it keeps things organized. And here, because we want to explore, I'll call it RMD, and it's a knitter file, so you should be able to basically just run this, and it might work. There's a lot of graphics in this one, so I'm going to actually stop this for now, and and run it line by line. So this is just due diligence at the beginning to get some of the past set, etc. Nothing too exciting here. You've seen it all before. Nothing interesting. Put this in. Okay, so and now we're going to read in our RDS file. So this is the one that we saved last time, and it's still version 1.0. So I'm just going to pull that back in, and yeah. So the one thing here is that the species start at around what is that? 55, 56, 57, 58. The column 59 up until column up until the end of the tibble, the columns of the tibbles are all species. And so for simplicity here, I'm going to set those numbers. Species start is 59, species end is the number of columns, and the number of species total is just the end minus the beginning. So the taxa are pretty long. Names are pretty long, right? This is this giant strings, characters. So what I'm going to do is I'm going to put them in a separate table, and I'm just going to label all of our taxon from 1 to 35,000. So for example here, if I do this, somebody's cutting their hedge on a Sunday in St. Lambert, which, I should call the police. This, silence is sacred in St. Lambert. Okay. Well, somebody once called the police on me on a Sunday for sledgehammering some concrete in the backyard. So now I feel like I should do the same. All right. So if I look at Tara now, what happens is that all the taxon are basically just like taxon 1 up to the end. No problems. Okay. And we can do some reality checks on these guys. For example, I could just plot, for example, the depth that a sequence is taken at across taxon 2. So if I do that, and I can see that here I have the depth, and these are the taxon. Man, what's going on? It is no, it's Monday. I guess it's Thanksgiving, so they're allowed to make noise. Huh. I thought Thanksgiving should be sacred somehow. But okay, so basically here you can see that there is a relationship, right? That taxon 2, a lot of it's often detected in shallow waters, and it's rarely detected in deeper waters. Okay, it's not a huge finding, but I'm not even sure what taxon 2 is. I could look it up by saying, okay, going back to my table, rig taxa names, and look at the second entry. And this is this proteobacteria, this Rhodospiralis. So yeah, well, it's mostly found in shallow waters, but sometimes in deeper waters. Well, what else could we ask? So is the distribution of species counts different across sites? What about Procolocaris? This is one of the most common organisms in the world, literally. It's all over the ocean. So I could do this, and I recommend that you stop here and you try to understand what I've done here with the statement grepel. basically I've tried to detect every taxon that was identified with the word Prochlorochorus in its name. Let's just take a look at the first guy, taxa 3, and plot that. Okay, and if I do that, Prochlorochorus. So the thing is that this is a photosynthetic organism, so it's doing photosynthesis. So you wouldn't expect this at depths very deep because you have no sunlight at that depth. I'm not sure where the mesoplastic, what is it, the mesodermic ocean? Is that how it's termed? Where the light stops, but I would think it's not more than 250 meters. And you detect a little bit, but it's mostly detected in shallow water. Okay. So what about depth and temperature, for example? So what if we asked, well, I don't think I need this, but what if we asked, what's the relationship between the depth of a sample and temperature? I would guess that you should see the deeper you go, the colder it should be. And what do we see is, in fact, that, right? That at the shallow profiles had higher temperatures, and it drops down. I would bet that these guys here were from the Arctic Ocean, those samples, because near the surface. And that of course could be checked by going back and looking for those points in the Tibble. What about salinity and temperature? What would you think? I would think that there's a pretty strong relationship. And here's a nice little thing called a 2D bin. So along this axis is the salinity measure. Along this axis is the temperature. And well, you know, to me, it doesn't seem that. I guess, you know, high temperatures you have more salinity. It's a little bit skewed that way. Certainly, low temperatures have, you know, relatively low salinity. I don't know what that sample is that has very low salinity and a very high temperature. It'd be interesting to figure out what that is. This point here, it means actually there's a lot of guys to count. There's a lot of different sites, almost maybe 15 across the Tara Ocean's data that had moderate, like middle salinity, but high temperature. I don't think it's the strongest example. And this should be, what about salinity and depth? I think, you know, that's an interesting question to ask too. If I put this in, well, that's maybe a bit stronger. It seems like, generally speaking, the salinity is lower at great depths. Maybe there's an exception there. There's quite a few Tara Ocean sites at that depth that have, well, it's relatively low. Well, I guess, no, I would say here it's definitely the case that, yeah, at great depths you have relatively low salinity. But yeah, this has very low salinity and it's at the surface. Well, okay, so I think it's really fun to look at those. So it's really easy for you to go and explore, and you have literally, like, you know, a couple of hundred variables to sort of look for associations. Okay, but as we said, we'd like to look at as many variables simultaneously as we could, but we have 35,000 taxa, so there's no way we're going to plot all of them at the same time. So we're going to have to choose which ones we're really interested in. A common way, or a common method many people use, is to choose the most variable guys across these 35,000 choices. And that's based on this measure of sample variance. Okay, so I put a link to a YouTube video explaining sample variance in the required reading. It's quite straightforward. And I also have a slide here of what the sample variance means. So suppose we looked at the distribution of counts like we did in the previous, a second ago. And each X here is one taxon. So we would measure for that taxon the distance from it to the sample mean, okay? And we would square it. Now, you don't really need to, why do we square it? It doesn't matter. But basically that's this. That's an individual taxon, and you subtract the sample mean from it and then square it. And you do it for all the points, from here to here, from this guy to here, from this guy to here, and on this side too. And you sum them all up. You divide by basically the number of points, and that's your sample variance. Okay, here's another distribution that's flatter and broader than this distribution. Will it have higher or lower variance? Well, again, for every point, you calculate the distance to the mean from this side too, and you square it and you add them all up. Because it's broader, the distance from the points, the length of these lines here, are going to generally be longer. And so the variance of this distribution that's flatter is going to be bigger than this distribution. The most variable guys are these points in the tails that are furthest from the mean. That's the guys that are, for example, not present in one data set, but present at 1 million copies in another, not data set, I'm sorry. What I meant to say was that there are organisms that were not present at 1 tera ocean sampling site, but then super prevalent at another site, and maybe moderately prevalent at a third, and fourth, heavily prevalent. It's really going back and forth, and that's the guys that are sort of like, they're the ones that are changing the most, right? So they're really interesting. Here's another distribution. So this is very tight around the mean. So the length of these bars in this distribution are all short. And so the variance of this distribution will be less than the variance of this distribution, which will be less than the distribution of the variance of this distribution. Okay, so what I do in this code here is basically compute the variance for each taxon, and I come in and I plot that with ggplot. That's just a distribution now of variances, okay? And then that should show up. Yeah, it looks a bit like what we had before, but now again you have lots of taxa that have very large variance. And now what I do is I arrange in descending order the variances, and I select the top 100. So I would recommend that you slowly go through that code. That's a good exercise for you, and there'll be some assignment questions for you to explore it. And that's basically the top 100. The most variable guy actually is this undefined. So that's really kind of a boring guy, right? The first guy of the most variable guys is this one that's the gutter. You know, when you go bowling and you miss the pins, you hit the gutter. That's what the undefined guys are. But other guys are really interesting here, like this Ocean Spiralis and this Brocodicia. These guys are pretty prevalent players in marine systems. So I'm going to make now a heat map across these 100 most variable taxa using the geome tile in ggplot. It makes a heat map. And in fact, I'm going to make an interactive heat map. So to start, what I'm going to do is I'm going to remove that taxa 1, which was like all undefined, okay? And what I'm going to, that just gets rid of that guy that's all undefined, guys. It's not interesting, right? And then I'm going to do some fancy pivots to get the matrix ready for ggplot, and do some other things that you could take a look at on your own time. I just want to show you graphically here. You'll have time on the assignment to explore that. And then I'm going to call, whoops, what happened there? Sorry. And then I'm going to call the ggplot function. And finally, I'm going to call this ggplotly. That turns the ggplot into an interactive tool. So when I do that, see here that, well, it looks pretty ugly. I have all these white columns in here, and can you take a guess at what those white columns might be? Well, they're basically, if you recall, at the join level, there was all those samples that didn't have sequencing data, and we see that now because they just end up as, if I scroll over these things with the nice ggplotly function, I can see that the count is NA. There was no measure done in that sample, Tara 068. So these white samples are all basically empty and should be removed. If I go to a red region, that's very high counts for those taxa. So this taxa in these samples are all very high, like 3 to 400 times that 16S rRNA was red. If I go to a blue region like here, they're around 2 or 3 or 4. This legend has to be updated. It didn't work out, so just ignore this for now. It's just to get a rough idea. But you see here, it's Tara Ocean samples by the taxa, right? For the top 100 most variable taxa. But it doesn't look good, so let's fix that first. So I'd come back here, and I could use this nifty function called drop NA. And that gets rid of any rows that are NA, like as you might imagine, right? So if I come in here, and now I'll just repeat this again, but now with that remove NA, oh my, copy and paste it in, and it's going to look a little bit better. Well, I think it looks a little bit better. It still looks chaotic, but those white columns have disappeared. But you know, we can see that some rows and some columns are kind of similar, but they're scattered throughout that that heat map. We're going to look not too long from now, in 3 or 4 lectures, at class discovery, and we're going to revisit this issue of clustering to get more information from these heat maps. For now, I just wanted to show you some nice, interesting ideas. First, if on your own or in the assignment perhaps, you can come back and explore this code. But what I'm doing here is I'm going to reorder the rows of this matrix by the feature. So in the table. So if I show you here in Tara, my Tara data structure, $feature, in fact I should do this. I should use the unique. Tara feature, there's different depths, or what they call, they pigeonhole the depths into different categories, like DCM for deep, and sometimes an oxygen minimum zone, surface water, or the mesopelagic, or the marine epipelagic, the mixed layer. So I'm going to reorder each of these Tara oceans into their 6 categories here, and this is what that code does. It basically gets that ready for, it reorders the factor. I can show you the heart of it. It's right here, that it's reordering the sample labels by the feature. And if I do that and I paste it, it looks a little bit better. I'm going to show you right now. You see here, now you see that all of these samples in here are kind of bluish, right? And you know, these are a little bit reddish, and these guys are a little bit more reddish, and then these guys in here are kind of sparse. But you can see if I walk along, all of these guys are DCM, see that? It's all the deep chlorophyll maximum layer. And then I switch. See, when I make the switch from DCM to mes for the mesopelagic, the pattern changes, right? This pattern is all mesopelagic, right? That's the mesopelagic, and it switches to the separate one, which is the mesopelagic and marine oxygen minimum. And then it switches into surface water, and the pattern changes, right? And then it's going to change again, okay? And then finally it changes. Oh, these are all surface, actually. All of these guys down here are surface water. And so you can see that the taxa that are picked up actually change depending on whether it's deep water or mesopelagic or surface, right? But still, it's not really optimal because these guys have not been ordered. And we can see that some of these rows are very similar to each other. And we're going to come back to this concept of hierarchical clustering. And I'm just going to show, the code here is to do it just for visualization. This code in here tries to recluster the rows to bring that out more, to bring out the relationships between the taxa, which taxa have similar behavior. And it's not that, it's a bit tricky, to be honest. It's a bit tricky. And you don't need to worry about that now, but just so that you see when I put this in and I view it, you can see that it actually starts to look quite nice. The Tara Oceans data is starting to say something. And here, it's not perfect, but you do see patterns now. Like these guys in here, all of these taxa in here, they have a pretty similar behavior all the way across this matrix, right, with each other. And down here, you see that they're not really, they're only identified at low levels, and then a few guys at very high levels in here in the mesopelagic zone. Just in this region are they identified. Other guys, you know, yeah, so now you can see that in both dimensions you start to see some interesting patterns. And although this is still far from perfect, yeah, these guys here are interesting. They're observed very low in the deep water, and these guys here in the mesopelagic are observed quite high, right? And then finally they're not observed at all at the surface waters. So, you know, they're different classes of organisms, right? And but they're related in where they're expressed, and so they're suggesting different patterns in different ecological zones. So, you know, hopefully that spurs your interest to examine this data set a little bit more. So this file is available to you too. Okay, you should take a look at the definition of variance. We're going to come back to that. You should get it out of the way now. But definitely here, I'm just providing you a bit of code so that you can explore. And so to end here, that's the end of the data wrangling for this data set. And well, we're going to come back and use this data set and the breast cancer transcriptomics. And next lecture, we're going to wrangle one more data set, a complete genome, into RStudio R. And so we're going to have those three major data sets to do a lot of computational biology on in the third part of the course. So I realize this is an incredibly technical lecture. I really recommend going slow through it. That's the wrangling process, but you know, we captured it well and we survived. So, okay, until next class.